Hello guys, I am Diptesh, currently studying BMLT in AIMS Kalani. So today we will be discussing about acid fast stain. So let's start. Okay, so acid fast stain. What is acid fast stain? So this is a stain that stains the acid fast organisms. So what is the meaning of acid fastness? Acid fastness is a physical property that helps certain bacteria, certain organisms to withstand the effect of acid. So if when we are putting acid, it can retain the primary stain and withstand the effect of acid. That's why it is acid fast stain. So acid fast stain was discovered by Paul Ehrlich and subsequently modified by Zil Nielsen. So that's why acid fast stain is also known as Zn stain. So full form of Zn, it is oftenly asked question that what is the full form of Zn in the Zn stain. So that is Zil Nielsen. This staining is done to identify acid fast organisms such as Mycobacterium tuberculosis and others. We will see after that what are the organisms that are acid fast. Acid fastness is due to the presence of mycolic acid in the cell wall. That mycolic acid is present in the cell wall of Mycobacterium species. So because of that mycolic acid, Mycobacterium species, so the acid fastness. Now coming to the procedure. Before coming to the Zn procedure, we have to make a smear. So how the smear is made? Smear measuring 2 into 3 centimeter in size is prepared in a clean grease free glass slide. When we are making it from the sputum, we have to choose the yellow purulent portion of the sputum. So smear should not be too thick, not too thin. How will you define that it is a perfect slide? When placed over a printed matter, the print should be readable through the smear. After making the smear, you have to heat fix it. Coagulation of the proteinaceous material in the sputum will facilitate the fixing of the smear. So this is the smear preparation. Here we are fixing the slide by heating it. So now come to the procedure. So in the procedure, we are using primary stain here, carbol fusion, 1% carbol fusion. 1% carbol fusion is poured for 5 minutes. Intermittent heating is done by flaming underneath of the slide until the vapor rises. So what is intermittent heating? Intermittent heating means we are not heating it like that is the stain that we have put in the smear that comes to boiling, not like that. Just slightly vapor will come from the stain, will stop heating. Heating helps in better penetration of the stain. We have to take care that the stain does not dry. After 5 minutes, we have to rinse the slide under the tap water. At this point, the smear on the slide look red in color. So what we are using as primary stain here, this is carbol fusion. Yes, this is intermittent heating. What is the meaning of intermittent heating? Pour carbol fusion on the fixed smear and apply heat beneath the slide until the vapor rises. Do not boil. We are not boiling it. That you have to keep in mind. Then comes decolorization. Decolorization by using 25% sulfuric acid. It is done by pouring 25% sulfuric acid over the slide allowing it to stand for 2 to 4 minutes. Then the slide is gently rinsed with the tap water and tilted to drain out extra water. Okay. Properly decolorized slide will appear pink in color. So if the slide is still red after that 2 to 5 minutes, if the slide is still red, we have to keep sulfuric acid for another 1 to 3 minutes, and again we have to wash it under the tap water. So this concentration of the sulfuric acid is very important as decolorizing agent. This is 25% sulfuric acid for decolorization. Now we are using counter stain. So 0.1% methylene blue is poured onto the slide and left for 30 seconds. After 30 seconds we will gently wash under the tap water and allow it to dry, and after drying we will see this in 100x, and how it will look like this. This background blue color is due to methylene blue, and these are the bacilli that are stained with acid fast stain. So how these are looking? This is looking like pink color. So this is a perfect Zn stain slide or acid fast stain slide look like. This shows the bacteria and the background is blue. Here it is the steps of the acid fast staining. We are putting carbol fission, then heating it, intermittent heating, then wash. After that we are using decolorization. That decolorizing agent is 25% H2SO4, then again water wash, then we are using methylene blue as a secondary stain, then wash, then air dried slide, and we are seeing it under 100x, and this is how a slide look like under 100x. So what will be the interpretation? The red color acid fast bacilli we can see. This is your acid fast bacilli, and the background, that is non-acid fast structures, will take the methylene blue color. This is a smear. Now come to the modifications of acid fast staining. Normally we are using hot method of Zn technique, that is commonly used. So here it is written that hot method of Zn stain is commonly used. There are some modifications, that is cold method. So cold method is known as Quinon's method. It is modification where the intermittent heating is not required, and it also differs from the normal Zn stain because heating is not required. Phenol concentration in carbol fusion is increased, and duration of carbol fusion in the stain is also more. What are the other modifications? We can use acid alcohol instead of H2SO4. We can use malachite green as a secondary stain. Yes, this is the chart. That's why I told that the concentration of sulfuric acid as decolorizing agent is very important. It is 25% for Mycobacterium tuberculosis, because Mycobacterium tuberculosis is very much acid fast, because the mycolic acid content is very much high in Mycobacterium tuberculosis. For Mycobacterium leprae we need 5% H2SO4. For Nocardia that is 0.5%, and acid fast parasites that are Cryptosporidium, Cyclospora, Cystoisospora. For that organisms we need concentration of 0.25 to 0.5%. So this is the reason why Mycobacterium shows acid fastness. If you can interpret that this is the peptidoglycan layer of cell wall present in the gram positive organisms. Besides that, acid fast bacteria contains mycolic acid layer. This is a lipid layer. This lipid layer binds with the primary stain and we told the primary stain. That's why when we are putting acid, decolorization is not occurring. What are the acid first organisms? Already told that mycobacteria species. Other than that, nocardia, parasitic oocysts. Some parasites like cryptosporidium, cyclospora, isospora, that are acid first that shows acid first properties. So these bacilli are of mycobacteria. Background is blue color and the bacilli is red in color, and those are the parasites, that is cryptosporidium. The background is blue and cryptosporidium stains the carbofucins, that is Zn positive. So this was today's discussion about Zn stain. This is a very routinely used stain. So what are the things you have to remember? Full form of Zn stain, why bacteria showing acid fastness, method of Zn stain, what are the procedure, what are the primary stain we are using, what is the decolorizing agent we are using, what is the counter stain, this is the process, and what are the modification. So that's it for today. Thank you.